morning. Last time when we were talking about the plants, we talked to you about the primary root system. Today what we want to do is to talk to you about the primary shoot system. You've already had some introduction to the shoot system, but I want to go ahead and just repeat a couple of the landmarks that we're going to be referring back to today. The first thing we want to keep in mind is that the shoot system is made up of a stem, which is divided, as we said, into nodes and internodes. And a node is, this should be review for you, is the place of the attachment of the leaves. So node is where the leaves are attached. We also said, and it's evident on here, that a leaf is part of the shoot system, and that we can divide the leaf into two parts, the blade and the portion which connects the blade to the stem itself, called the petiole. And there was one other thing that we mentioned last time. We said that in the axil, we would have axillary buds. In the axil, we would have axillary buds, and that these could grow into branches. There are only two other tissue types or organ types we need to add to this picture to get the shoot system. And those two other types, which we're going to discuss at more length a little later in the class, are flowers and fruits. Taken together, flowers, fruits, the stem with its nodes and internodes, and the leaf with its blade and petiole, and the axillary bud, all of these constitute the primary plant body. That is, they are all products of the apical meristem. And this, of course, would be the shoot apical meristem, because we're talking about the shoot system. This shoot apical meristem, we said, is located at the tip of a branch. Shoot apical meristem is located at the tip of the branch. And the shoot apical meristem makes derivatives, or you could say new cells, which then elongate and contribute to the increase in length of the stem. So in a branch or a shoot system like this, it would have an apical meristem at its tip, and as the apical meristem produces new derivatives, which then elongate, essentially those new derivatives push away the apical meristem and elevate it into the air, and thereby increase the length of the shoot. Now, because the apical meristem is so important, it is the place where, for our discussion for the beginning, it is the place where all the new cell types in the primary plant body are initiated. What I want to do is to spend some time with you talking about the shoot apical meristem. And this will have more relevance to you in a few more lectures when we talk about hormone physiology in plants and how it affects development, because the development is affected as the hormones interact with the apical meristem. So let's first talk about an apical meristem and understand that elongation growth is necessary for the stem to grow upward. Every branch which is produced and has an elongating axis has an apical meristem on it. And if the apical meristem is removed from the branch, it stops elongating, and it will no longer get any great distance until a new apical meristem is produced. So make sure you understand every elongating axis has to have an apical meristem. Now let's spend some time looking at the apical meristem, and for this discussion what I want to do for you is to make a section essentially parallel to the stem axis, and this section is going to cut right through the middle of the apical meristem, and because it is a long section or parallel to the axis, we call it a longitudinal section. Or as it is abbreviated, it's an LS. So we're going to make a longitudinal section of the shoot apical meristem, and we'll magnify it, and I'm also going to show you some slides of it. But when we make our initial views of the shoot apical meristem, at the very tip, we find what looks like a rounded dome-like structure. Dome-like structure at the very tip of the apical meristem. And on either side of the dome-like structure are bumps or protuberances. These bumps or protuberances are the origins of new leaves. And they're given a special name because they're the structure which originates to form the leaf. They're called a leaf primordium. A leaf primordium. So here we have a apical dome with leaf primordia on either side in this particular drawing of the apical dome. And this apical dome now has a region of cell division. A region of cell division. It is making new cells, and as we mentioned before, as a result, those cells elongate, and they are elevating the shoot apical meristem. Now, as the shoot apical meristem moves away from the leaf primordia that they have initiated, those leaf primordia begin to get larger and to mature. So further down on the axis, we might find an older leaf primordium. An older leaf primordium. And on this side as well, we find an older leaf primordium further down on the axis. Also at the shoot apical meristem, we find one other structure that you've already been introduced to that is produced as a result of the cell division activity of the apical meristem. And that is in the axil of the leaf primordia, we have a bud being produced. And we're going to return to this bud, but keep in mind that the buds grow out to give you branches. They give you branches. And you should already be set and confident with the conclusion that if these branches are going to grow out from the buds, and each branch is going to continue to elongate, that in fact each branch has to have an apical meristem on it. So ultimately, the buds have to develop an apical meristem. Very important concept. Now, let's take this shoot system which we have here, and instead of making a longitudinal section through the shoot system, let's make a cross-section through the shoot system. And let's look to see what kinds of cells, once they differentiate, are being produced as a result of the cell division activity in the apical meristem. These first cells that are produced are uncommitted. They don't have any particular cell type. But as the apical meristem moves away from these cells, they begin to differentiate into the various cell types which we have spoken of already. And let's begin by looking a little further down on this stem here. After the apical meristem has moved away, we're going to make a cross-section of a eudicot stem. Cross-section of a eudicot stem, and see what kinds of tissues have been produced as a result of the activities of the apical meristem. So this is a cross-section of the stem, and there are only a couple of cell types that we need to put on here. What we notice is that there are regions on the stem, and this is just a typical one I'm drawing here, which appear to have They appear to be distinct bundles. That's the name that's applied to them. A bundle. They're different from the surrounding tissue. And inside each bundle, what we find is the vascular tissue. So each bundle is comprised of the vascular tissue. And the vascular tissue is not randomly scattered in the bundle, but has a very precise orientation, with the phloem toward the outside of the bundle and the xylem to the inside. Now we have only a few other tissue types that we want to put on here. In the very center of the stem are parenchyma-like cells. This is given the name pith. Surrounding on the outside, again, the outermost layer, you've seen this before, is the epidermis. And then we have to add a new layer onto this, and this new layer occurs between the bundle and can be of various thicknesses, the bundle and the epidermis. You've seen this layer before in the roots. This is the cortex. So this is the typical cross-sectional stem structure of a eudicot. Now what I'd like to do is to compare that cross-sectional structure to the cross-section of a monocot stem. And I'm going to put the screen down now, but we're going to not look at it yet. So let's make a cross-section of a monocot stem and compare it to that eudicot stem. Again, it would be round. We're just making a cross-section of it, someplace below the apical meristem of the monocot stem. And rather than having distinct bundles, which appear to be slightly below the epidermis, what we find in a monocot stem is that the bundles appear to be randomly scattered. So these are bundles, and they appear to be randomly scattered. We also have an epidermis, just like you saw before. This is the shoot system of a monocot stem. And we're unable to distinguish a cortex and a pith because the bundles are so randomly scattered. And so this tissue in the center and all over around the bundles is just called ground tissue. Now, the bundles are something you're going to see in lab, and I'm going to show you a picture of them in a moment. And the reason I want to draw attention to these bundles is that if you look at the bundle, there's xylem and phloem, just like we saw before in the bundle, xylem and phloem. But what is particularly interesting and what will be very useful for you to see is that the phloem in the bundles is probably the best phloem in the whole plant kingdom for being able to see sieve elements and companion cells. So you can nicely see sieve elements, and you're going to get a chance, and companion cells. So these are the tissues that are produced as a result of the apical meristem activity in a stem which is showing primary growth. Let's look now at some pictures of the apical meristem and of the cross-sectional stem activity. Okay, this first slide, maybe, it says screen blank. Okay, there we go. This first slide is going to be of a shoot apical meristem in which we've taken off a few large leaves, but we still have some leaf primordia at the very end of the apical meristem. And now if we continue to remove the leaf primordia and get down to the very tip of the shoot, here we have the apical dome sticking up, and these are some leaf primordia, the newest leaf primordia which have been produced. And this apex has cell division activity occurring in this region, which will cause the apex to elevate, leaving behind these leaf primordia and leaving a space for new leaf primordia to initiate. Now, if we make a longitudinal section through that apex, we can see some of these tissues. Here is the apical dome, that structure we talked about before. Here would be the region of high cell division activity. Here are new leaves coming off, leaf primordia, which are been produced. And in the axils of those leaf primordia are the buds, the things that are going to become the branches. If we make a cross-section now of the stem of a eudicot, we can see a couple of the elements I wanted to point out to you before, and you'll see them. In the very center of the stem is the pith. These red cells are the xylem, which you know as being tracheas and vessels. Here is the phloem, the cortex, and the epidermis, the same ones you've seen before. Here we have a cross-section of a monocot stem, and here there's no distinction between pith and cortex. The bundles are scattered, and if we look at the bundles in a little higher power, we can see that they have these large elements. These large elements are vessels, and you're going to see them in the lab. And in the higher power view, you can actually hear the vessels we showed you before. You can actually see the phloem. And what I want to point out to you is these large and small cells. The small cells are the companion cells, and the large cells are the sieve elements. So this is the great example of the phloem. Now, the apical meristem not only initiates new cell types, but it also initiates new cell types in a very precise arrangement, a very precise order. No cell, no tissue in the plant appears randomly. So through the activities of these cells which make up the apical meristem, we have a very precise arrangement of all the plant parts. And trying to understand the controls for this very precise arrangement is currently an area of intense study. If you look at a branch like this, for example, let's ask about the leaves. If you look at a branch like this, it's hard to see any precise arrangement to the leaves. They may, to your mind, be inserted randomly. But if we could collapse all of these leaves down to one plane, one single plane, what you would find is that these leaves actually do have a precise pattern. And what I want to do is to show you a slide in which that exact thing has been done. The leaves have all been collapsed down into one plane, and what you can see, each of these structures is a leaf here. What you can see is that the leaves are arranged in spirals. In one case, the spirals run counterclockwise, and in another case, the spirals run clockwise. So you can see a series of spirals that make up the arrangement, in this case, of the leaves. But these spirals, in fact, apply to all the tissues that are initiated at the apex. Now, people have been very interested in studying these particular tissues, and what I would like to do is, first of all, to show you what was one of the first observations or conclusions that were made from studying shoot apices and asking about the arrangement of the leaves. And what was discovered is that in plants which had spirals, one spiral running in one direction, that you could find some plants with only one spiral running in another direction. In some plants, you would find one spiral running in one direction, counterclockwise, and two spirals in the other direction. In some plants, you could find two spirals running clockwise and three spirals counterclockwise. What would be the next number in the series? 5. And how about after that? 8. So most of you recognize this as the Fibonacci series. Plant parts are arranged on the plant and can be described in their arrangement by the Fibonacci series. We don't understand how this Fibonacci series arrangement comes about. We don't understand the genetics of it. But it can be used as a way of explaining the type of arrangement and characterizing the type of arrangement of all plant parts. And indeed, much of life outside the plant kingdom also has their parts arranged according to a Fibonacci series. Let me show you a few more examples now of plants which demonstrate nicely the Fibonacci series. Here is an agave, which has a very complicated Fibonacci series in which you have a certain number of spirals running clockwise and a certain number running counterclockwise. And if we make a cross-section through the shoot apex of this particular plant, what we can find is that if we look at the spirals, we would have spirals running in the counterclockwise direction, and we could have spirals running in the clockwise direction. So here's a clockwise direction spiral. Now, if you look at the difference between these and running in the counterclockwise direction, the difference between 22 and 35 is 13, 35 and 48 is 13. So one of the numbers is 13. In the other direction, the difference in the number would be 41 to 49 is 8. So this apex, this plant, is described in the Fibonacci series as 8 plus 13. 8 spirals in the clockwise direction and 13 in the counterclockwise direction. Again, we don't understand the genetics of this, but we know that it tells us that the arrangement of the parts isn't random, that there's some underlying program which is influencing the arrangement of the parts. And this Fibonacci series can be applied to many different organs, many different tissues. Here is a pine cone similar to what you've seen in the lab. And again, you can see spirals running in this direction and spirals running in this direction. Again, arranged in a Fibonacci series. If you look at a flower head like a daisy flower at the very center, or a sunflower, you can see a certain number of spirals running clockwise and a certain number running counterclockwise. And even in something as common as a pineapple, where we can look at it, you can see, and I have one up here in front if somebody would like to check, or next time you go to the store, a certain number of spirals running clockwise, a counterclockwise, and a certain number running clockwise. And to make this a little more evident, here you can put pins in, and you can actually see the number of spirals. Oh, looks like the program quit. Well, that's okay. That was perfect. Okay. Let me just get back to our program here. I don't know why I quit. Shoot system. So they'll give you a chance to get notes. All right. So there's the pineapple. Okay, so this is the Fibonacci series, and this particular series demonstrates to us that the plant parts are arranged in a very, very precise pattern, and that this pattern governs the development of the plant, yet we really don't have much of an understanding of how this pattern works. Okay, let's now continue to talk about the shoot system. And what I want to do is to talk to you about what is probably the most variable organ on the shoot system, with the exception of the flower. The flower is certainly the most variable tissue or organ type that you will see on the plant, but the one that comes closest to it in its variability is the leaf. And so what we want to do now is to talk about the leaf. Now, leaves are primarily designed for photosynthesis. But the capacity of the leaves to capture light is also, and their form to capture light, is also influenced by the environment in which the leaf is growing. And you may recall back to the discussion where we talked about the hairs on leaves. Those are structures which have been put onto the leaf to influence the amount of light which gets to the leaf surface. So leaves are designed for photosynthesis, but they can also undergo many modifications to allow them to adapt to particular environments. And I'd like to begin our discussion of leaves by taking a typical eudicot leaf and looking at a few of the cells inside the leaf, the anatomy of the leaf, because this is going to help us to understand how the leaf can perform, as you will see, so many different functions. So here is a leaf that I'm taking off the stem here, a leaf, and what I'm going to do is make a section, a cross-section of the leaf, and we're going to look at this section through this, and we also have a cross-section of a leaf down here with this model, so to get your orientation right. So let's make a cross-section of a eudicot leaf. And we're going to begin with two cell layers that you are familiar with already. That is the outermost cell layer, which is the epidermis. Epidermis. And there's an epidermis on the top and the bottom of the leaf, as you might expect, because all of these cells have to be covered on their outside. Now, if we look directly beneath the epidermis on a typical leaf, we see these elongated cells. They kind of look like salamis hanging in a butcher window. These are the site of most photosynthesis. Site of most photosynthesis. That's where most of the sunlight is captured. And we call these cells the palisade, and then the next word you know already, parenchyma. Directly below these cells is a large region of cells which look like they're very loosely packed. These cells have lots of wall area, lots of surface area, and that's why they're loosely packed. And this wall or surface area is important because this is where CO2 is absorbed. Now, how does the CO2 get into the leaf? We mentioned last time that there were pores. And we said that this pore was called stomata. So CO2 would enter. It would get absorbed or taken in by the cells which are loosely packed. And these cells are called spongy parenchyma. And then using the sunlight captured in the palisade parenchyma, we would make sugars and starches. Now, there's one other type of cell or region we have to indicate on this cross-section of the leaf. After the sugars are made, they have to be moved away. They have to be transported away. So what I want to do is briefly talk about the vascular system in the leaf. Everybody knows the vascular system in the leaf. These are sometimes called by you veins. Again, going back to the old terminology of the 19th century where people applied the same sort of name to plants as they used for animal cells, which had the same function. Now, in a eudicot, the veins are net-like in their patterning. This is a eudicot. In a monocot, the veins are parallel. The veins are parallel. And this parallelness of the veins allows you without, with great security to say whether or not a plant is a monocot or a dicot. It isn't a hard and fast rule, but it's a good general rule if you have a plant that its veins, if they're parallel, those plants are probably monocots. These veins, probably the one that is most familiar to you, and if you look at a typical eudicot leaf, there's one big vein which runs down the center of a eudicot leaf. This is known as the midrib, or the most prominent vein on the leaf. In a monocot, if you go outside and look at the monocot leaves, you won't find any prominent veins, any midribs. Now, what I'd like to do is to mention to you the fact that although leaves are primarily designed for photosynthesis, in evolution, they have evolved to become other structures. And I want to mention a couple of those structures to you. In this plant which we have over here, and I hope some of you come up to see it, this is known as the blackhorn acacia. If you look at the leaves, the green part of the plant, and I'll just draw one leaf up here, the leaf is divided, almost looks fern-like in its appearance. But what I want to draw your attention to is at the edge of each of the teeny leaflets which make up this big leaf, there are these kind of golden bodies, which are about 0.5 millimeter or less in size. These golden bodies are called Beltian bodies, after the man who discovered them. And again, you don't have to write this down. This is kind of for your own sort of satisfaction, information, and wonder. Beltian bodies. And they are rich in nutrients. And let me tell you the story now which happens. This plant grows in Mexico, Central America, and the plant is inhabited by a quite vicious ant. This ant harvests the Beltian bodies and uses the Beltian bodies for food and takes the Beltian bodies back to their homes. Where are their homes? Their homes are inside these rather large thorns or horns on this particular plant. So this plant is giving these ants a home and food. What is the plant getting out of it? Well, these ants are so vicious that if an animal comes over and tries to graze this plant, the ants will go out and attack the animal, And if another plant should overtop this plant and shade it and actually reduce the amount of sunlight reaching this plant, the ants will go to the other plant and chop down its leaves. So these ants and this plant have evolved together. This is a form of coevolution, and you might ask yourself, how did something like this possibly evolve? How did it evolve to have these nutrient-rich Belgian bodies on the edge or the ends of the leaves? Another modification of a leaf is one that I have up here in front, and this is a leaf of a plant that this is a plant actually where you can see its leaves, which is known as a window plant or a stone plant. Maybe some of you have these at home as your garden plants. A window plant. They're sometimes called stone plants because they look like stones. And if we look at these plants, which come from South Africa and which are very endangered in their environment, here is the soil top. And these plants consist of two leaves, which are mostly buried in the ground, below the ground. And let me tell you how these leaves have become modified. So each of this is a leaf. The photosynthetic surface of the plant is along this edge of a leaf. So this is the photosynthetic area. It's along the edge of the leaf. Now these plants grow in areas where they are predated by animals that eat them, and so they are called stone plants because they have evolved to look like the stones in the soil that they're growing in. But beyond that, they have another very unique adaptive mechanism. And that mechanism is they grow in areas not only where there are predators who would like to eat the plants, but also in areas where the sunlight is very bright. And as we mentioned before when we talked about the plants growing high in the Andes, too much sunlight can destroy the photosynthetic mechanism. So how do these plants deal with this high sunlight? And this is a really remarkable way that they do it. At noontime, when the sun is most intense and highest, in order for the sunlight to get to the photosynthetic surface, it has to enter through the windows, through the pores, as you'll see if you look at these plants, and I'll show you a picture of them in a moment. Enters through the window. So let's take a beam of sunlight. And before it can get to the photosynthetic surface, it has to travel through this sort of non-green tissue, which makes up most of the leaf. And in traveling through this non-green tissue, its intensity is reduced. So by the time it gets to the photosynthetic surface, it's the right intensity for photosynthesis. Now how about the end of the day, where you know the sunlight is weaker and not as quite intense? Let's put the sun over here at the end of the day so that it's beginning to set. The plant still wants to use the sunlight, but if it decreases its intensity too much, it won't serve photosynthesis. So in this particular case, when the beam comes in, it only has to travel through a small amount of tissue to reach the photosynthetic surface and thereby be able to do photosynthesis at the optimal rate. So this is a remarkable passive system for keeping the light at the right intensity. And again, for some of you who are thinking about being bioengineers or interested in biomimicry, the term I gave you before, here's an interesting example of how you can control sunlight intensity passively. You're not doing any mechanics at all in this particular system. Finally, before I show you some pictures of leaves, I want to just bring up this leaf here that many of you have seen or you might have as a houseplant. This is a leaf of a philodendron or a monstera plant. This leaf has holes in it, and this plant, if you see it growing in its native environment, actually climbs up other trees. It grows high in the forest canopy. So one of the questions people have raised is, what do you think the purpose of the holes are in these leaves? These holes develop actually by a method that many of you have heard of before, by programmed cell death. Cells in the leaf primordium are programmed to die to make these holes. So does anybody have any thoughts about what these holes might do functionally? Any ideas? Yes? Right. That's a really good suggestion. So at high levels where they are in the trees, it's very windy. It might be a way of keeping the wind from destroying the blade, which is so big here. Another suggestion which has been made is that these plants make these holes because when an insect or an animal is looking either to predate a leaf or to lay its eggs, they will see the holes on this leaf and think it's damaged and thereby not be a good place to lay their eggs. So this could also be a protective function from predation or from pathologies from other animals laying their eggs on the leaf. Just an idea that hasn't been examined, but certainly worthwhile understanding. Now, leaves come in a tremendous number of shapes and sizes. And up here in front, I have for you a palm leaf. This is not a typical leaf, but to give you an idea of how much of the biomass of the plant can be invested in leaves, you've got this palm leaf, and you will see, compared to leaves of very small plants, which is one of the first slides I'll show you, that the change in size of leaves can be orders of magnitude. And again, they are primarily designed for photosynthesis, but they also are designed to be able to meet environmental constraints and take advantage of various aspects of the environment. So let's look now at a couple of pictures of leaves. This first slide is a cross-section of the leaf of a eudicot, and I want to point out to you a number of things which we have seen. Here we have the upper and the lower epidermis. Here is that mid-vein, that prominent vein in the leaf. And if we look at this leaf in a little higher power, here's the upper epidermis, the lower epidermis. Notice this is the pore through which the CO2 enters. This would be a stomatal complex. Here are the salamis hanging in the window. This is where most of the photosynthesis would go on, the palisade parenchyma, and then directly beneath it is the spongy parenchyma. And here you can see the CO2 would come in and be absorbed by these cell walls or cell surfaces. To give you again a scale of the leaf, here is a small leaf compared to a leaf like we have up here in front, orders of magnitude bigger. This is a leaf of a plant which is mainly tropical, although there is a plant of this growing in Berkeley. This particular plant is supposedly the plant that Buddha sat under when he was able to come to his great meditation and conclusions, and is a plant which belongs to the fig family, which we've talked about before. And because of its association with Buddha, it's called ficus, because of the fig family, religiosa. But the reason I bring this up is because this plant has a modification of the leaf which is called a drip tip, and it is meant to channel the water, rainwater, to the root system. Here is our plant, the monstera plant, or the philodendron with the holes that we talked about before. Sometimes leaves are modified, just like roots are modified. In this particular case, the leaf has been modified into a tendril to allow the plant to climb. And this is pretty obvious in something like a pea plant, where you can modify the leaf and you can then have a grasping organ. Sometimes leaves are modified for water storage. Here is an example of a succulent in which the leaves fill up with water and allow the plant to overcome long periods of dryness. This is a plant which grows in the desert southwest, and it has these thorns on it, or these protective structures, which you can see better here. These protective structures actually are petioles, the base of the leaf in which the blade has fallen off, and the petiole itself has been turned into a thorn, allowing for some protection from the plant. Here is a view of the lithops, and again, you can see these. We're looking down on top of it. Here are the two leaves, and these darker structures are the windows. Here is another leaf where the darker structures are the windows. We're looking down on top of it, and again, there has to be a diffusion of the sunlight through varying lengths of tissue for photosynthesis to occur. Prime examples of modifications of leaves, probably the best examples, are found in the insectivorous plants. In the Venus flytrap, the modified structure which captures the insect is a leaf, and with proper stimulation, the leaf can close and digest the insect. The sundew plant, in the sundew plant, the modified structure which captures the insect is the leaf. The leaf secretes both glue to capture the insect, as shown in this slide here, and also enzymes to digest the insect. And for those of you from California, especially Northern California, this is our native California pitcher plant. It's called Darlingtonia, and it functions by actually luring the insect in, and the insect then is digested at the base of this modified leaf. Here is a leaf that we would find from a plant called nepenthes in the tropics, and you'll probably get a chance to see them when you go up to the botanical garden. These leaves are large enough that people have actually found rats and small mammals inside, and they weren't actively captured. What probably happened is that the mammal or rat saw some insects down here and tried to get the insects, and then fell in and drowned in the water and is being digested. Here are the blackhorn acacias, which we have before. Here you can see the sort of fern-like leaves. And these golden bodies, which I'll show you a little better picture here, these golden bodies are the Beltian bodies that the ants harvest. Here you can see some pictures of the ants actually going along and taking off these Beltian bodies, which they then carry back to their nests or their lair. And here are the horns, where the ants have hollowed out and they have a home, so they got food and a home. And finally, I just want to show you a few pictures of leaves that some of you may have seen. In these particular leaves of water plants, the blade is very large, and the petiole anchors the plant or the leaf to the water. You can see how big they are. And in Victorian times, before there was Facebook and YouTube and so forth, one of the pastimes was to float children out onto leaves and see how long your child could stay there before it gave up. It was kind of a contest between people. Okay. In the last few minutes of the talk, we've mentioned that leaves can be modified and leaves are part of the shoot system. What I'd like to do is to tell you about modifications of the actual stem of the plant as well, and how these modifications are used by humans and are used to benefit the plant depending upon what environment it grows into. So what I want to talk about, first of all, are stem modifications. And for this talk, I want to draw your attention to really two types of modifications of a stem. One modification is for the storage of nutrients. And the nutrients which we want to talk about are starch and sugar. And the other modification is for the storage of water. Let's talk about the situation about the storage for nutrients. Probably the prime example for the storage of nutrients that most of you are familiar with is a potato. A potato is a stem which grows underground, and this potato is a type of stem which is called a tuber. Well, if this is a stem, and we said that a stem had nodes and internodes, where are the nodes and internodes on a potato? In fact, the nodes can easily be found on a potato because each node is indicated by where there's an eye of the potato. And for those of you who leave potatoes sitting too long in the dark and a shoot grows out, essentially that eye is an axillary bud growing out into a new shoot. Where are the leaves then, if this is a node on a potato? Well, this is a structure which develops underground. It really doesn't have a need for leaves for photosynthesis. But if you looked at the development of this tuber, you would actually find some structures which are very, very tiny vestigial leaves. So if this is a stem, and if it has nodes and internodes, it should follow the Fibonacci series. And the answer is yes, it does. And I'm going to show you a picture of how the buds, or the eyes as you know they are called, are arranged on this structure, which is full of starch and which is a modified stem. Another example of a modified stem that many of you from East Asian cultures may be familiar with is taro. Taro is a structure which is known as a corm, a modified stem, and there is starch stored in this corm. It is of very high quality, and it is often used to feed children, babies, because of the high quality and the small nature of the starch. Taro, a modified stem which is known as a corm. Now here is an interesting stem which both has modifications of the stem and of the leaf. In this particular case, what has happened in this stem is that the internodes now have not elongated. So we have a situation in the stem in which the internodes have not elongated, and in the onion, I'll write this for you, no internode elongation, and more importantly, the reason onions are good for us to eat is because sugars and sometimes starches are stored in enlarged leaves. So if we could make a longitudinal section through this onion, we would find all of the internodes, all of the leaves, probably attached at what appear to be the same point. There would be no elongation of the internodes. This theme of a lack of internode elongation is also familiar to you in a plant such as celery. In celery, each of these petioles represents the base of a leaf, each of the structures which you eat is celery. In celery, the internodes have not elongated, and so all of these petioles appear to be attached to the stem at one point. In fact, there's a small distance between them, but in general, you could say that there's little to no internode elongation in something like this particular stem. And an example that many of you are familiar with that has no internode elongation is this cabbage. Cabbage is also a modified stem in which the internodes don't elongate. And if we cut this cabbage down the middle and look at the attachment of the leaves, what we can find, and I'll leave this up here and I'm going to show you a slide of this, is we can find the stem axis, this white part running up the middle. The leaves are attached to the stem axis, but there's been very little internode elongation. So in a cabbage, what we have is a stem which has not elongated to make this structure. For those of you who buy Brussels sprouts, a Brussels sprout is essentially a small cabbage, an axillary bud on a stem which has not elongated. Let me now show you some slides of these examples, and then I'll let you go. Okay. So we'll go ahead and do the slides. Okay. Here's an example of a plant in which the stem has been modified to store water. And I have a cactus up here in front for those of you who want to be convinced about that it's a stem structure. And this particular cactus, the stem can shrink or increase depending upon the availability of water and serves a very important function adaptively. Another interesting situation with regard to how stems have adapted and changed, this is a cactus which is known as a caterpillar cactus. The cactus essentially lays flat on the ground, and it grows at its tip over here. Apical meristem is located here. And as it grows, the back part of the cactus dies. And so the plant actually moves from one area to the other, in contrast to what we've said about the life history of most plants. Here is a very interesting cactus in the tropics in which the stem of the cactus now, rather than just being solely for support, actually functions to grasp onto some other organ, which in this case is somebody else's tree, and elevates the cactus into the air. It actually grows on other plants. Here is our tuber, and this structure, which is known as a bulb, which is the onion, B-U-L-B, another modified stem. And what I'd like to point out to you, as seen in the next slide, is if you draw the Fibonacci, or you draw the lines through the axillary buds or the eyes, you can actually see the Fibonacci series on the potato. And finally, I just want to show you a picture of the cabbage. The cabbage is a stem in which the internodes have not elongated. And if we take the stem and we cut it down through the middle, you can see where the shoot tip would be up here at the top, where the apical meristem would be. And then these are the leaves which are coming off. And there's very little internodal elongation between the leaves and between adjacent leaves. So this is a plant which has nodes and internodes, but very little internode elongation. Okay, have a nice weekend, and I'll see you on Monday, I hope.